Hello, good afternoon, everyone. So welcome to today's session of Global IMMUNOTALKs, and it is my great pleasure to host Nicolas Manel today. So Nicolas did a PhD in virology in Montpellier in France, looking at cell entry by HTLV and defining entry receptors. And he then moved to NYU in the US, where he carried out a postdoc with Dan Littman. And amongst other things, he looked at innate immune sensing of HIV by dendritic cells. He returned to France in 2010 and he set up an independent lab at Institut Curie, where he is now the equivalent of a full professor. And his lab has sort of gradually moved from innate sensing of retroviruses to more generally innate recognition of nucleic acids, not only during viral infection, but also in cancer and in aging, as well as the applications of this knowledge for therapy. So Nicolas has made key discoveries on how viral capsids can shelter retroviral DNA from cGAS recognition, how viruses can transport the seeds of their own destruction in the form of packaged cGAM that activates cGAS in the target cell, and also how self-DNA can trigger cGAS in various instances, from virus infection all the way to cancer and aging. So in a minute, I'm going to ask Nicolas to deliver his lecture. But as usual in this series, I'm going to start with a personal question before the talk. And Nicolas, your question is, how do you prioritize which research questions to pursue in a rapidly evolving scientific landscape?
First of all, thank you very much, Caetano, for the very kind introduction. Thanks to all the organizers and everyone listening to this today. I think we prioritize our research first based on our results and what we think is interesting. On top of that, we're constantly asking if what we're doing is important, and we are asking this at every step to go back to the central question of importance. And we're also trying to figure out if we have an edge, a sort of original approach to the problem we're trying to ask. I think what we're trying to do is not do the obvious, I could say, not do what maybe other labs would be doing. We're trying to find a way to do it in an original and creative way.
Well, that's very good. So we're very much looking forward to your seminar, Nicolas, in which you will show us actually how you do this, non-derivative and non-research. And the talk of Nicolas Manel's talk today, the title, I should say, is Centromeres Reimagined in the Immune System. So thank you, Nicolas.
Yeah, thanks, Caetano. So what I will do today in the 45 minutes or so of the talk is first describe what we do in general context of immunology, then get to centromere biology and how we arrived at this from the perspective of innate immunity. I would then go in a bit more detail in a recent study that we put out to explain how the molecular mechanisms are working. And at the end, I will give a sort of vignette in opening and trying to open new directions in these themes, and hopefully overall convince you that centromeres are a very exciting part of our genome and that we need to rethink them for the immune system. Before getting into that, let me highlight and encourage all of you to participate and submit an abstract for the upcoming pathogen immunity and signaling meeting that I'm co-organizing and will occur in a beautiful place called the Naturalis in Leiden in Netherlands. That will be in June. We will have short talks selected on abstracts and poster prizes. The deadline for abstracts is February 1st. Right, so our lab is interested in central question of immunology, which is self and non-self discrimination. And we came to this question from initially the angle of detecting viruses inside cells. And for example, we are asking how is non-self detected inside cells, what maintains intracellular tolerance to self. And over the years, the field and us realized that the same pathways that are involved in this detection of viruses, pathogens, are overlapping with pathways that get deregulated in aging. And therefore, we're also asking questions such as how is self being altered during aging, and what are the cell intrinsic drivers of aging? So these questions are organized. I want to showcase the lab here, organized around 4 axes with a wonderful team that I love to work with every day. We have a group working on antiviral sensors, how viruses are detected. Another group working on the regulation of the cGAS STING pathway at the molecular cellular level. A growing group working on biology of aging, and finally, a group working on application of this to immunotherapy. Now, coming back to the initial question, and what I want to discuss today is how cells detect viruses, particularly in the nucleus. Why the nucleus would be a challenging compartment? Well, this is because nuclear DNA viruses, such as herpes viruses, and retroviruses enter the nucleus and cause genome instability. But of course, there is an excess of self-DNA and RNA in the nucleus, challenging the conventional understanding of self-non-self discrimination based on solely nucleic acids. The central pathway that we are interested in to ask this conceptual question is the cGAS-cGAM STING pathway of innate immunity, which is now recognized as a universal pathway of cytosolic DNA detection with both infectious and non-infectious functions. In the pathway, pathogens or steroid damage release DNA, initially thought to be cytosolic DNA with a cytosolic DNA sensor, cGAS. cGAS, an enzyme, produces a second messenger, cGAMP, which is cyclic GMP AMP, that then has a receptor in cells called STING, and upon binding to cGAM, STING translocate in the cell and transduces signals through transcription factors such as IRF3 and NF-kappa B for antiviral and immune activation. So this pathway is now recognized to be central to protection in a number of viral conditions, and also central to both natural and therapeutic anti-tumor immunity. But being such a powerful, responsive, reactive system, it is also when chronic conditions are engaged, it is also involved in pathogenesis of multiple diseases, a growing list of diseases shown here. But this is to say that this is an essential pathway, an important pathway for inflammation and understanding immunity. As I mentioned, the sensor and the location of DNA was initially thought to take place in the cytosol, and it certainly does so. But I want to highlight here the notion that the receptor itself, cGAS, is also, if not in larger amount, present in the nucleus than in the cytosol. I'm showing you 2 movies here to illustrate this. Those are mechanisms of cGAS entry into the nucleus. At the top is a dividing HeLa cell with GFP cGAS in green here. And at the onset of mitosis, upon nuclear envelope breakdown, cGAS coats the chromatin and remains associated with the chromatin at the next interface. And another mechanism here of nuclear cGAS entry into the nucleus is shown here in non-cycling cells. These are human monocyte-derived dendritic cells in which we express GFP cGAS again in green. And this particular dendritic cell is going through a narrow space, a constriction of a few micrometers, which represent the space, that the dimensions that these cells encounter in vivo. And this is done in collaboration with the Piel lab at Curie Institute. And what you can see here is upon a passage of the nucleus through the constriction, cGAS that was initially cytosolic enters the nucleus. So this is due to transient nuclear envelope ruptures in interface. The nuclear envelope bursts open and then repairs, allowing rapid entry of cGAS into the nucleus. These are only illustrations for the purpose of the talk, just to give you the image that cGAS has also a life in the nucleus. Therefore, it must be tightly regulated because it is a highly reactive protein and there's an excess of DNA, as I alluded to already. And indeed, there are plenty of mechanisms that have been worked out already to explain how cGAS would be kept in check in the nucleus. The major mechanism is tethering to histones, which limits activation of cGAS. Other mechanisms include inhibition by BAF at the nuclear envelope and a specific mechanism also of degradation of cGAS. So we are not addressing this question. We are rather interested in addressing the mirror question, which is if the sensor is in the nucleus, what is it doing there? Is it involved in pathogen recognition? And if so, in which context and how would it be regulated? We had already done one contribution to this question by showing that HIV-2, so this is a related virus to HIV-1. HIV-1 is much less pathogenic and drives a more potent immune response. And we demonstrated that HIV-2 is detected in the nucleus, but by cGAS, but not only by cGAS. This recognition is first dependent on an interaction between a protein called NONO and the viral capsid protein, and only if NONO, that is in complex with cGAS, recognize the viral capsid, can then cGAS detect the viral DNA and drive an innate immune activation. So we were interested from here in asking, are there other situations where viral sensing would be mediated by nuclear cGAS, perhaps implicating viral proteins? And to get to this, we started by asking, where is cGAS located in the nucleus? Is it randomly associated with chromatin, or is it perhaps enriched somewhere? And if it is enriched, what are these enrichments telling us? So to do this, we did ChIP-seq-like approaches to sequence the DNA that was associated to cGAS in the nucleus, both in human dendritic cells and mouse dendritic cells. And what was really striking is that there was an enrichment for so-called satellite DNA in both human and mouse ChIP-seq data sets, suggesting a conservation in evolution of this enrichment. Of course, cGAS coats chromatin everywhere. I'm not saying that this is exclusive to other parts of the genome. We do see cGAS covering all chromatin, but we're measuring relative enrichment here. So to confirm this in a kind of orthogonal manner and also understand what are the satellites, we looked at metaphase spreads. So these are metaphasic chromosomes, because the satellites are the DNA sequences that are located at centromeres. And I will come back to centromeres. Centromeres are these constrictions of chromosomes here. This is the sequence or the satellite repeats, and they can also be marked here by a protein called SNP. And we could indeed quantify that there was an enrichment of cGAS at the centromere on this metaphasic chromosome spread, and this was quantified here. So cGAS is partially enriched on centromere, at centromere. Okay, so from here I will spend a couple of slides to give you an introduction of what we know about the centromere and what is relevant for this talk. So showing you here the human centromere. It's obviously the structure of the genome that is essential for chromosomal segregation in mitosis, because this is where microtubule attach the kinetochore. The centromere is defined epigenetically. It is defined by a protein, SNP. SNP is a histone H3 analog. So in the nucleosome, it replaces histone H3. And when there is SNP, this identifies the centromere. So SNP is located on the centromeric DNA, but it's not alone there. There's a second protein called SNB. SNB is a DNA binding protein that recognizes centromeric sequences. and it provides a genetic memory to preserve centromere position. Together, they recruit CENT-C, and CENT-C then works sort of like a scaffold to assemble the rest of the centromere protein complex. At the genetic level, at the DNA level, centromeres are composed by highly repetitive sequences that are in human called alpha satellite array. But it is not the DNA sequence that defines the centromere, it's the position of CENT-B. So for an immunologist, what I would like to propose is that it's interesting to think about centromeres as one approach to define the inheritance of self. Let's take a functional centromere, and we have a mother cell with normal centromeres that will undergo genome replication, duplication, and mitosis. And during mitosis, chromosomes will be properly segregated thanks to the functional centromere. And this will lead to daughter cells. And in these daughter cells, the self is preserved through generation. If we imagine now the situation of a dysfunctional centromere, so centromere in red that for some reason is not working properly, in mitosis, this centromere will not be correctly attached to microtubule and will not be correctly segregated, creating imbalance and differences in terms of chromosomal segregation in the daughter cells. And conceptually, we could view this as an alteration of self-identity. The initial identity of the mother cells is not propagated correctly through generation. To try to convince you that this may be real, I will first start by quoting Angelica Amon, who said, "It's not true if you cannot show it in yeast," and highlight this paper from the Barral lab in Zurich, who showed that this is true, and in the case of yeast. This is an example where using plasmid DNA, it is the presence of centromere sequences on the centromeric DNA that defines whether the plasmid DNA, circular DNA, will be correctly segregated to daughter cells and maintain identity. Or in the absence of proper centromere, it will be retained in the mother cell. Okay, so a bit more on centromeres. So they are very interesting, unique structure. They're highly divergent and unstable DNA structures. Here, I'm taking a picture from a review from the Malik lab showing that centromeres are highly diverse across species. These are various chromosomes in blue from various species, fungi, Drosophila, plants, animals. And the centromeres are highlighted in red. And what is very striking is that across evolution, the configuration of the centromeres has varied extensively, going from single points to spreading the full chromosome as if there was a centromere along the entire chromosomes. The centromeres are also highly unstable in cells. This is a recent work, for example, here from the Esashi lab, showing that centromeric DNA, the part of centromeres on our chromosome, are probably among the most unstable part of DNA sequences. They seem to break all the time spontaneously. So this was sort of term centromere paradox, because all eukaryotes need centromeres for chromosome segregation. But at the same time, it is a rapidly evolving and highly unstable molecular entity. Now, to give a little bit more example of the relevance of centromeres thinking about viruses, I want to highlight a few cases. Starting by saying that this represents 3% of the genome, and that there is an interplay of this sequencing with viruses. So first, herpes simplex virus 1, and I will come back to that. This virus establishes latency in neurons, and in neurons, the virus positions its DNA close to centromeres. This is also true for cytomegalovirus as a similar mechanism of positioning its genome close to centromeres. HIV patients, so let's consider elite controllers, patients that control the viral replication. In these patients, there is still the presence of a reservoir of virus, the virus is not randomly integrated in the genome. There is an enrichment in centromeric DNA in these patients. This is also true in or visible in evolution. So for example, here, an endogenous retrovirus called ERF-K, one of the ERF-K, has expanded in the evolution from primates to humans, and there are more than 100 copies of a single provirus that has invaded the human centromeres. And last example is SNB itself. So the key protein, the second key protein for centromere identity and positioning. The origin of SNB stems from a protein from a transposable element tiger, suggesting an ancient history relationship between transposable element viruses and centromeres. And finally, I think what really unlocked in the recent years the research of centromere is the following: that this was a blind spot of the genome, of the human genome, and that is now unveiled thanks to telomere genome sequencing. This opened the gate to study the centromeric DNA in human and now in other species. Indeed, recently the full human centromeres have been mapped and sequenced. This has revealed, for example, highly original mechanisms of evolution, from younger sequencing, pushing back older sequences. And also the high abundance of transposable element in alpha satellite repeats at centromeres. So again, highlighting this notion that there is an interplay with virus-like sequences. All right, so with this in mind, I will now go into a specific role of centromeres in viral detection. And we started this by knowing that cGAS was enriched at centromeres, asking if there was a candidate virus that could perhaps be interplaying with this positioning of cGAS at centromeres. I mentioned already herpes simplex virus 1, HSV1. It is a nuclear DNA virus. It has a so-called lytic infection and a latent infection. In both cases, central protein for genome replication and maintenance is this viral protein called ICP0. But first, let me show you that indeed the virus is viral DNA is in proximity to centromeres. These are low-resolution images from neurons in latently infected mice, and while the viral DNA is not overlapping with telomeric sequences, the viral DNA here is overlapping with minor satellites, which are the centromeric sequences in mice. The virus puts in DNA in proximity to centromere. And it does that with ICP0. So ICP0 is a remodeler of the proteome in infected cells. It is a sumo-targeted ubiquitin ligase, and it is a large protein, more than 700 amino acids, and it has already been implicated in a number of activities during viral replication in the control of intrinsic immunity and innate immunity. Now, where is ICP0 in cells? Its localization changes during various target cells conditions. But for example, here, I want to highlight that ICP0 was early on observed to localize at centromeres. So this is staining for the ICP0 protein in dividing cells, and the dots are the centromeres where ICP0 is located. So the protein ICP0 goes to centromere, has the capacity to go to centromere. What is it doing to centromeres? It, being a ubiquitin ligase, it leads to degradation of centromeric proteins. So, for example, here an ICP0 expressing cell during infection, CENP-C is degraded, CENP-A is degraded, CENP-B is degraded, and this depends on the ubiquitin ligase activity as the mutant is not degraded. So it remodels the centromeres, and from these pictures, we can only see degradation of centromeric proteins. So we wanted to ask, since ICP0 does that, is this perturbation doing anything to innate immune activation? So we asked if ICP0 perturbing centromeres would trigger a response in human dendritic cells. This was the work of Xavier Lai in the lab, starting from primary blood monocytes, differentiating them into dendritic cells, and simultaneously expressing vectors to induce ICP0. So first, we confirm that the ICP0 protein leads to degradation, perturbation of centromeres. This is a CENP-B staining here, and indeed, the cells that were transduced with ICP0, monocyte-derived dendritic cells, degrade CENP-B in the presence of ICP0. And this was quantified here. Okay, so is this doing anything to innate immunity? And a simple way to test this is to measure activation markers in human dendritic cells, which is what we're showing here with CD86 and Siglec-1 as surface markers to reveal activation of an immune response. And indeed, it was very striking that expressing ICP0, the protein, was sufficient to induce a response from the dendritic cells with upregulation of these markers. And this was dependent on the ubiquitin ligase activity because ICP0 mutant was not doing this upregulation. Now, is cGAS implicated in this? We here knock down cGAS in this experiment and ask what is the impact on the response. And indeed, cGAS is implicated in this recognition, so to speak, of ICP0, because when we knock down cGAS, we have a reduction in CD86, Siglec-1 is reduced, interferon lambda 1, that is secreted protein, is reduced. Interferon beta secretion is also reduced. There is a remaining interferon beta production that is cGAS independent, that is due to a second pathway, the MORC3 pathway, that is totally independent from this pathway. If cGAS is implicated, we wanted to then determine if it was active and where was it active. Was it active in the nucleus? And we resorted to measuring cGAMP, the second messenger, in cells expressing ICP0. But before doing that, we did a control, which is to transfect DNA. And if we transfect DNA in human dendritic cells, we can measure cGAMP production, as you can see here in log scale. And this production is enriched in cytoplasmic over nuclear fraction, consistent with cytosolic detection of DNA. When we express ICP0, we also observed induction of cGAMP production by these cells. But interestingly, contrastingly from DNA, the cGAMP was accumulating in the nucleus, suggesting that there was activation consistent with activation of nuclear cGAS. So let me repeat this. Essentially, we have a viral protein that activates a DNA sensor in the nucleus. So how could this work? This is essentially what I showed you so far, that ICP0 through a mechanism activates cGAS, leading to an activation of the pathway with a parallel activation of just interferon beta through the MORC3 pathway identified by Moritz Geit and Russell Bunce, but the two are really independent. The next question was, what activates cGAS? And I will highlight two pieces of data to illustrate this. We find out that there is an amplification of centromeric DNA, that it is the centromeric DNA that is responsible for cGAS activation. So let me show you one approach here we did. We decided to quantify the abundance of the genomic DNA in these cells expressing ICP0. Because of the complexity of the repeat sequence at centromeres, we used a mapping-free approach using k-mers, so with tiling sequences across the genome. And we then analyzed the differential abundance of k-mers between ICP0 and control. And we were able to show that k-mers mapping to centromeres were highly differentially abundant, increased in the ICP0 expressing conditions, while other k-mers at chromosome arms or in the mitochondrial genome did not show this amplification, this increase. Now, this was extremely surprising to us because this occurred in monocyte-derived cells that are quiescent in G0, and we verified this. There is no cell cycle related DNA replication occurring in these cells. But to convince you that there is indeed an amplification of centromeric DNA and to show you where is this DNA in the cell, I want to share here an orthogonal method where we did imaging of centromeric DNA. This is an immunofluorescence fish experiment where we stained the centromeres with CENP-B and we quantified the abundance of centromeric DNA with a fish probe. And doing this again in ICP0 expressing cells was very clear that there was an amplification of centromeric DNA detected by the FISH probes. And in addition here, that this centromeric DNA remained local. It was not diffusing in the nucleus or elsewhere in the cell, really remained local at centromere. We quantified this across donors and cell unique cells, and because ICP0 degrades SEN-B, there was an anti-correlation. Less SEN-B was associated with an increase in abundance of alpha satellite DNA. This was also true over multiple donors. So this is what I showed you so far, that ICP0, this viral protein, leads to amplification of centromeric DNA, and it is then the centromeric DNA that can presumably serve to activate cGAS. So how is, the next question here was, what amplifies this centromeric DNA? What is the mechanism independent from the cell cycle? By analyzing the targets of ICP0 as a ubiquitin ligase, we figured that a genome stability control pathway was involved, which led us to propose or to test the involvement of a specific type of DNA replication machinery called the translesion DNA synthesis. What are these enzymes? They are specialized DNA polymerases that have unique active sites capable of accommodating damaged or distorted template. However, this comes at a cost. Unlike conventional DNA polymerases, like the alpha, they have reduced fidelity. They are mutagenic polymerases. And they are recruited by ubiquitination, typically when there is a lesion and the conventional polymerases have difficulty to continue the replication. TLS is recruited and then activated and is able to continue synthesis. Why is this important at centromere? So recently, our collaborator on studying centromeres, Daniele Facchinetti from the Institute, showed that this TLS machinery has a non-redundant role at centromeres during the cell cycle. So just for one slide, let's just put our heads into a cycling cell. Now let's imagine a cycling cell and what would be the problem with centromeric DNA. And the problem is the repetitive nature of centromeric DNA, which creates structural hurdles for a DNA replication, hairpins, misfiring, misalignment, mishybridization, which essentially gives a hard time to DNA pol alpha and the conventional DNA polymerases at replicating the centromere. And if this is not properly processed, centromeres break and there are rearrangement occurring. So the cells have evolved a way to prevent this kind of breakage at centromeres by having the TLS machinery, the translesion synthesis, to help replicate this difficult template during DNA replication. So this is shown here. One key data is here. So this is measuring from this study the speed of DNA replication by either control or cells in which the translesion synthesis TLS has been suppressed. And so if the velocity in bulk across the genome is measured, there is no difference. Most of the genome is replicated without the TLS. But the centromere replication, centromeric DNA replication requires the TLS. If TLS is inhibited, the speed of replication is much slower. Therefore, we decided to imagine that maybe, even though we're not in the cell cycle, the TLS might be the machinery involved in centromeric DNA amplification due to ICP0 expression and ubiquitination by ICP0. Here, I want to show you first that we tested this with an inhibitor of the TLS that had been recently described, GHRE06. And first, let me show you that indeed, when we block the TLS with this inhibitor, we abrogate the centromeric DNA amplification. This is shown here by qPCR with primers against alpha satellite centromeric consensus, or primers relatively specific to chromosome 4 or chromosome 10. And ICP0, as you can see, induces this centromeric DNA amplification, but co-treatment with the TLS inhibitor abrogated this amplification, indicating that indeed the amplification was occurring through this machinery. Would this then block cGAS activation downstream? The answer is yes. Indeed, blocking the TLS with these compounds abrogated the induction of lambda interferon shown here, and also cGAMP production. Now, this was not a problem at the level of cGAS itself, because when we instead transfected cells with exogenous DNA and treated with the same inhibitors, cGAS was functioning normally. The level of cGAMP was similar, so cGAS was active, and the level of interferon was also similar. So it was inhibiting specifically the ICP0 response and not a general effect on cGAS. To confirm this with another method, we generated knockouts, and also to highlight approaches we perform in the lab. In primary monocyte dendritic cells, we knock out the various genes coding for TLS-related proteins, as well as the polymerase alpha gene, which is not expected to play a role here. And indeed, we were able to confirm that knocking out these polymerases prevented interferon lambda 1 induction by ICP0, while knocking out the polymerase alpha gene had no effect on interferon lambda production, suggesting specific involvement of the TLS machinery. And correlating with this, we also saw that centromeric DNA amplification was abrogated, reduced by the knockout of the TLS, but not by knockout of the pol alpha. All right, so the mechanism, and I didn't show everything, but the scenario is the following. ICP0 triggers through ubiquitination a reactivation of the translesion synthesis pathway outside of the cell cycle at centromeres, which amplifies locally centromeric DNA, leading to cGAS activation, cGAM production, innate immune activation. All right, this is interesting, but this is all based on overexpression of viral protein. Is this mechanism relevant during a viral infection with the true virus? Here we did the following reasoning. If this activation of cGAS, an antiviral pathway, by ICP0 is important during infection to trigger such antiviral response, then it would make sense that a successful virus, such as HSV1, has already evolved a way to counteract it, and that this pathway is actually suppressed in the wild-type virus, enabling the virus to evade immunity and be so successful. Turns out that, indeed, HSV encodes a suppressor of the translesion synthesis, UL36USP, another protein. UL36USP is a so-called, let's call it, non-canonical open reading frame identified by the IDEPLO lab in one of the other proteins of the virus. And more recently, the Zheng lab showed that this UL36USP, which is a D-ubiquitinase, so it opposes what ICP0 does, suppresses translesion synthesis. It prevents translesion synthesis activation. We then decided to test if indeed that was what was going on, that the wild-type virus would evade the pathway because of UL36USP. And to test this, we collaborated with the lab of Lars Dolken to test viruses mutated at a single amino acid in UL36USP, so cysteine 65 to alanine, and tested their impact on centromeric DNA amplification and innate immune activation. So I'm showing you first the centromeric DNA amplification again, reminding you it's in dendritic cells, there is no cell cycle. The wild-type virus, indeed, does not trigger this centromeric DNA amplification. It is evaded by the wild-type virus. But mutating the D-ubiquitinase, UL36USP, now reveals the pathway. We can now see amplification of centromeric DNA, and this amplification is dependent on TLS, transcription synthesis, because it can be blocked by the inhibitor. Is this matching with innate immune activation? The answer is yes, but let me guide you through this because it's a little bit complex scenario here. We're measuring cGAMP, which is the downstream of cGAS, and we're infecting cells with the wild type or the TLS, the virus carrying the mutation, focusing on the TLS pathway. If we take a wild type herpes simplex virus, of course, yes, this virus will activate cGAS. This is presumably the incoming viral DNA. This is very well established by James Chen and others that the infection by HSV1 wild type does trigger cGAS through the viral DNA, and we can certainly see that. Now, what happens is that if now we restore the TLS pathway activation on top of this initial amount of cGAMP, we see an increased abundance of cGAMP that is now produced, that is attributed to centromeric DNA amplification because, again, it's blocked by the inhibitor. So we estimate that about a third of cGAS activation can be restored when this pathway is revealed. So we would like to propose that this is a new antiviral system in the nucleus. Why such system would be required? Because unlike the cytosol, the nucleus is highly concentrated in self-nucleic acids, challenging the direct detection of nucleic acids. And instead, it seems that cells can detect the presence of viral proteins that manipulate genome integrity. And this fits very well with the concept of effector-triggered immunity, where it's the effect of viral proteins that is detected. We term this mechanism, this antiviral system, VICR, for viral-induced centromeric DNA amplification and recognition. I explain in detail how this system is working for HSV1. We also showed that a similar system exists for cytomegalovirus, where IE1, the protein of CMV that tethers the genome to centromeres, trigger a similar centromeric DNA amplification, leading to cGAS activation. So why did we call this VICR? This is because in the Roman Empire, the vicarius, the vicar, served as a second in command to a specific superior. So this is depicted here. If we imagine a virus expressing proteins, manipulating the cell, the vicar represented here is the second in command who detects these changes and provides now centromeric DNA amplification. And it is the centromeric DNA amplification that is detected by the superior here. So what are the perspectives to this mechanism? First, we think that it may represent a function, so to speak, of centromeres outside of mitosis. We know that biology does not like to maintain things unused. And it is, if you think about it, it's hard to believe that centromeres would be used only when cells divide, when we know very well that cells spend most of their time in tissue not dividing. Another perspective is that centromere instability, here induced by viral proteins, may be relevant in inflammation and aging through the pathway that we uncover. And going back to this picture, what I want to highlight here is that we uncovered another case of pathogen recognition in the nucleus by way of cGAS. In this case, a viral protein is detected by cGAS by way of an intermediate, which is self-DNA amplification by the TLS. And to finish, I want to highlight a vignette. So we were thinking, if viral proteins can drive centromere instability, what else is out there? What can we uncover that may be unknown for the moment? And we decided to ask, are there other situations of centromere instability in the immune system that we can detect? So before I show you the data, I want to refresh your memory about the centromere organization of the human centromere. Cent-A is this histone H3 variant that provides the epigenetic identity of the centromere with replacing H3 at nucleosomes. CEN-B provides a genetic memory. It's a second protein that binds centromeres and help restoring CEN-A, and then CEN-C assembles the complex. And to explore this instability in the immune system, this is the work of Elena Izquierdo in the lab in collaboration with the Daniele Facchinetti lab. We decided to start by measuring the level of these centromere proteins in this case, in immune cells from human blood. And I'm showing you CD4 T cells here from human blood, total CD4 T cells, and where we stain using intracellular staining, CEN-B or CEN-A. CEN-B does not show in this graph here very striking change in expression. It seems to be pretty well expressed. However, what was very striking was CEN-P. We did not expect that there would be a distribution of CEN-P, because in cycling cells, CEN-P is very highly expressed homogeneously. Instead, in human blood, it seems that there is a heterogeneity in CEN-P, and cells, a fraction of cells are essentially CEN-P low or perhaps CEN-P negative. So the centromere is not homogeneous in blood cells. There is a diversity, and we're trying to understand what is the cause and consequences of this heterogeneity. But to convince you that this is not just total protein expression, but truly an alteration of centromere identity, we also did imaging here. We sorted the cells and performed immunofluorescence. And this was with the help of Marie Dumont in the Daniele Facchinetti's lab. CEN-B here is stained to localize centromeres, and CEN-P here is quantified. And in CEN-P high cells, CEN-P is, as we would expect, properly located at centromeres. But matching the CEN-P low state, there was indeed a depletion of CEN-P from centromeres that we otherwise identify by CEN-B. And this was quantified here. So we're very excited now to decipher this centromere biology in the system. And I will finish with this slide, and hoping that I convince you that the centromere is not simply, as we learned in the textbook, a static structural element for chromosomal segregation. We have to reimagine the centromere, particularly in the immune system. It is, in fact, dynamically unstable as an element in our genome, and not only in mitosis, also in quiescence in non-dividing cells. Now, why is this particularly relevant in the immune system? Well, because we know that genome instability in immune cells is a key driver of aging, and in models has been shown to be sufficient to drive organismal aging. And therefore, we are very interested now, very keen in addressing the following question: is this reimagination of the centromeres in the immune system contributing to genome instability with mechanisms relevant to aging? And we're excited that we are going to be able to do this research in collaboration with Elsa Logareño's lab and Daniele Facchinetti's lab at Institut Curie. I will stop here. Thank you for your attention. I think I thank the various people who did the work during the presentation, and all the people who contributed to the work. And I look forward to addressing your questions afterwards.
Well, thank you very much, Nicolas, for this fabulous seminar. Now I'll remind all of the audience that the questions are asked on the YouTube comments section, where Nicolas' lecture will then be posted. You have a QR code up on your screen. So it just remains for me to remind you that next week's lecture will be given by Lang-Siew Yap, and I hope you will all be able to tune in. And so just thank you to Nicolas Manel once more for today's lecture, and thank you all for supporting Global IMMUNOTALKS.
Thanks, everyone. Bye.